I recently moved to the Bay Area. What I've learned so far is that if you want to make something happen, you do it from the garage. So let's get into genome biology. It's fascinating, at least for me, to realize that strings of 4 simple building blocks form the basis of every living thing on Earth. And large-scale measurements have improved sequence annotation, the what is what of DNA. And the synthesis of DNA has become affordable, even for small labs. And we actually think you need just 1 million base pairs of DNA to make a living cell. In letters, that's not even half of the famous textbook, Molecular Biology of the Cell by Alberts and colleagues. So we're looking at this really exciting convergence of opportunities. And in fact, we're already programming biological systems with groundbreaking applications in biotech and in health tech, but always at this limited scale. So a single gene or small interaction networks. But if we can synthesize whole genomes, we can really start to push the limits of our imagination. But unfortunately, fitting biology into the rules of exact science or into this exact science template to engineer robust systems is not really straightforward. The problem is that programming systems needs rules. And in the context of the genome, these are the answers to questions like where does what go? Which letters of the DNA can we safely exchange? And we actually do know a lot about the general design principles that govern genome biology from contributions from the labs of George Church, Pamela Silver, and Jason Chin, to name just a few. But sometimes a single point mutation can wipe out the function of a gene. We see this even in certain diseases in humans. And deriving rules per specific gene takes an immense amount of time or a very large collection of PhD students. I mean, think about how you need to change and test every single base in even a small genome of a couple million base pairs to know if exchanging them with a different DNA letter will have a detrimental effect or not. So whole genome engineering requires an approach to probe regulation, not base by base and gene by gene, but at scale. And Beat Christen at ETH Zurich set out to do just that. He took the essential genes of a freshwater bacterium, he rewrote them to enable affordable synthesis and genome assembly, and then strung them one after the other into a new genome. And he didn't know all design principles, gene-specific rules, and exchange over 100,000 base pairs letters of this genome. So you kind of expect some of these genes to not work, right? And what is the most straightforward way to test that? You let the cell decide. So we plugged these genes back into the cell in a piecemeal fashion and measured how the cell interprets them by RNA sequencing. So we compared transcription between native and rewritten genes. And I want to briefly highlight our findings relating to a specific gene which codes for a component of the ribosome. This is the protein translation machinery that we have in our cells too. The gene is called RPLS, and we trace its malfunction in the cell back to a regulatory hotspot of RNA bases involved in an RNA structure. And we think this structure guides the ribosome to translate RPLS mRNA into protein. So we conclude that we cannot exchange these specific bases. And we not only improve annotation of the native genome of Caulobacter, this freshwater bacterium that we based the rewritten genome on, but when we zoom out, we can actually extrapolate this gene-specific rule to other genes in the essential genome. So we translated our findings into what we call design principle 4. So taken together, we formulated general design principles that allow to improve the design of synthetic genomes. And summarizing it, we rewrote a genome, we synthesized and assembled it, we tested it in a piecemeal fashion in the native cell to learn how to do it better in the next round. And this design cycle can be run even with limited resources. And each measurement brings us closer to that final set of rules that allows us to build whole genomes with novel functions. So is genome design complicated? Yes. Are we there yet? No. Should you start to empty out that garage? Definitely. I also learned that everyone loves a good yard sale. So this work is based on research in the group of Beat Christen at ETH Zurich in Switzerland, and would not have been possible without the support from ETH Zurich and the Swiss National Science Foundation. Thank you very much for your attention.